In this chapter, we'll take a closer look at the properties and functions of macromolecules, and we'll spend most of our time discussing proteins, which is the category of macromolecules we'll discuss first. When we think of proteins, many of us immediately think of enzymes, but proteins play many different roles in cells. They help transport material into and out of cells. They're important for cell shape and cell motility, and there are even proteins that allow cells to defend themselves. And these are just a few of the types of proteins that are essential to the function of our cells. The monomers of proteins are the amino acids, and there are 20 naturally occurring amino acids that are used in protein synthesis. However, other amino acids exist in cells that are not used for the synthesis of proteins, and there are even amino acids that are created by modifying the 20 amino acids used in polypeptide synthesis once the amino acid has been incorporated into the polypeptide chain. All amino acids have the same basic structure. They consist of a central carbon, which is known as the alpha carbon. Attached to that carbon, it's a hydrogen atom, a carboxyl group, which is negatively charged, an amino group, which is positively charged, and these charges exist at physiological pH, and a side chain or R group. The side chain or R group is different for each amino acid. So this is what gives different amino acids their different properties, since the rest of the amino acid is the same for all 20. Since a sequence of amino acids of a polypeptide determines its shape, no two proteins can have the same sequence. In other words, an amino acid sequence will result in a specific three-dimensional shape. However, there is a really important exception to this that we'll discuss in the next lecture. 19 of the 20 amino acids occur as stereoisomers. Of the 20 amino acids, glycine is the only one that does not occur as a stereoisomer, since it does not have 4 different groups attached. You can see that 2 of the constituents attached to the alpha carbon are hydrogen. So this does not have those 4 different groups that are required to have a chiral or asymmetric carbon. An amino acid that may have confused you is proline, because you can see that the side chain or R group for proline is actually attached to the amino group. However, this amino acid still has 4 different constituents attached to the alpha carbon, so it can exist in 2 different configurations. Although amino acids exist as stereoisomers, only L-amino acids are used in protein synthesis in cells, though the reason for that is not fully known. One possible explanation is that selection for L-amino acids over D-amino acids occurred by chance early in evolution, and that this outcome became fixed to minimize the energy requirements needed for the maintenance of multiple stereospecific enzymes. I should note that D-amino acids are present in short peptides that are found in the cell wall of bacteria. Based on the properties of their R groups, amino acids can be grouped into different types. Your textbook groups them into 2 major types, hydrophobic and hydrophilic. And within the hydrophilic, there are charged and uncharged amino acids. As you can see by looking at the structure of the amino acids shown on this slide, you can tell that these are hydrophobic because they contain side chains that are largely nonpolar. And I need to emphasize that what we're looking at here is the R groups, because as you can see, the rest of the molecule is the same for all amino acids. Now, what are the characteristics that tell us that these side chains are nonpolar? Well, you can see that they mostly consist of carbon and hydrogen, and bonds between carbon and hydrogen are nonpolar because these atoms have very similar electronegativities. So we see here that these amino acids generally lack oxygen and nitrogen, in particular oxygen and nitrogen bonded to hydrogen, which would make that a polar bond. So these amino acids, because they are nonpolar, they're unable to interact with each other through ionic bonds, and they're unable to interact with water. For proteins that are present in the aqueous environment of the cell, hydrophobic amino acids are usually located within the core of the protein, away from the surrounding water, and they interact with each other through van der Waals forces and hydrophobic interaction. In this simulation, hydrophobic amino acids are depicted in brown, polar uncharged amino acids are depicted in green, and charged amino acids are shown in either pink or purple. Watch as the hydrophobic amino acids move toward the core of the protein. In transmembrane proteins, which are proteins that contain segments that pass through the membrane, hydrophobic amino acids will be found within the hydrophobic core of the lipid bilayer. Although a few amino acids in this section can be hydrophilic, and these are represented here in blue, most, as you can see, are hydrophobic, and these are the ones shown in green and in yellow. The other group of amino acids are the hydrophilic amino acids, and within these we have the polar uncharged and the polar charged. We can tell that these amino acids are hydrophilic because they are either charged or because they have functional groups that have highly electronegative atoms like oxygen bonded to hydrogen. Of the charged amino acids, aspartate and glutamate behave as weak acids due to the presence of a carboxyl group in their side chains. And those carboxyl groups have a tendency to donate protons. On the other hand, lysine, arginine, and histidine behave as weak bases since they have amino groups that have a tendency to accept protons. Of the polar uncharged amino acids, cysteine is a special case as it is classified as polar in some textbooks and as nonpolar in others, and that's because the difference in electronegativity between sulfur and hydrogen is small. Nevertheless, this difference, which is 0.4, is significant enough to consider the sulfur-hydrogen bond, which is a sulfohydryl group, a polar bond. As you saw in the protein folding simulation, polar amino acids, both charged and uncharged, tend to be localized to the surface of cytosolic proteins where they interact with water. Additionally, the charged amino acids are commonly found within the active sites of enzymes. More specifically, these amino acids are usually directly involved in the process of catalysis because they're more reactive than the rest. Now, this graph shows the results of a study that characterized the amino acid distribution within enzymes. The x-axis shows the different amino acids using the one-letter code for those amino acids, and the y-axis shows the frequency. In green, we see the frequency of the amino acid in the entire protein, in blue within the active site, and in red, those amino acids that are part of the catalytic site. So within the active site, the amino acids that are directly involved in the catalysis. The charged amino acids here are the last 6: lysine, arginine, histidine, aspartate, and glutamate. As you can see, these are the most common catalytic amino acids. To summarize, most proteins contain both hydrophobic and hydrophilic amino acids. They're amphipathic. In membrane proteins, the amino acids present in the cytosol and those present in the extracellular space tend to be hydrophilic, while those in the region that passes through the membrane, known as a transmembrane segment, tend to be hydrophobic. For non-membrane proteins, most polar amino acids are localized to the surface of the protein, and the nonpolar residues tend to be present within the core of the protein. Now, when I use the word residue, I simply mean amino acid residue, because that's the name given to an amino acid once it's incorporated into a polypeptide. Finally, an important thing to keep in mind is that there may be polar amino acids that project into nonpolar regions. In other words, we could have some polar amino acids within the core of a protein, as seen in the enzyme active sites. And this is related to the fact that hydrophilic amino acids, particularly those that are charged, are more reactive. As you saw in the question, you're expected to be able to identify the properties of the amino acids when provided their structures, and based on their property, decide where you would expect to find a peptide within a folded protein. I'll give you the amino acid structures as well as the names, 1-letter code, and 3-letter code, so you don't have to memorize that information. This short peptide contains the amino acids serine, alanine, asparagine, threonine, aspartate, and arginine. Of these, the only one that is hydrophobic is alanine, since it contains a methyl group as its R group. And since a methyl group consists of a carbon bonded to 3 hydrogens, these bonds are nonpolar. If we look at the other amino acids, we see that serine and threonine have hydroxyl groups, which consist of an oxygen bonded to a hydrogen, so that is a polar group. Asparagine is also polar as it contains an amide, which also contains electronegative atoms bonded to less electronegative atoms, so these are also polar bonds. Finally, we see that aspartate and arginine are charged, so these are also polar. So based on this, we can conclude that this segment of the polypeptide is most likely to be localized either in the extracellular loops of a transmembrane protein, in the cytosolic loops of a transmembrane protein, or at the surface of a cytosolic protein. Based on its polarity, this segment is not likely to be found within the transmembrane region of a polypeptide, nor within the core of a cytosolic protein, where we would expect to see more hydrophobic amino acids. Amino acids are linked together by a type of amide bond, which is a carbon-nitrogen bond, which is known as a peptide bond. As you can see, this bond results from a condensation or dehydration synthesis reaction, where a water molecule is released as a bond forms between the carbon of the carboxyl group of one amino acid and the nitrogen of the amino group of the next amino acid. Once an amino acid is part of a peptide, we refer to the amino acid as a residue, as I mentioned earlier. As you can see, the charges of the amino and carboxyl group are no longer present after the formation of the peptide bond. However, as we'll see later, even though this carbonyl group and this amino group are part of the peptide backbone and they are not charged, they're actually very important for protein structure. As a consequence of the way in which polypeptides are synthesized, their ends are different. So this type of macromolecule has directionality. On one end, there is an amino group, so this is known as the N-terminus, and on the other end, there is a carboxyl group, so this is known as the carboxyl terminus or C-terminus. Amino acids are synthesized from N to C, meaning that as an amino acid comes into a growing polypeptide chain, it attaches to the carboxyl group of the previous amino acid. So the last amino acid that is incorporated into the chain will have an exposed carboxyl group. The immediate product of translation are the polypeptides, and once these fold and acquire biological activity, they're considered proteins. Nevertheless, those terms are used interchangeably. Proteins can be monomeric or multimeric. As the name suggests, a monomeric protein is composed of a single polypeptide, while a multimeric protein is composed of more than one polypeptide. And we'll return to this point later when we talk about the different levels of protein structure.